Monitoring. Regular monitoring of the farm is key for determining its Prrs epidemiological status and consequently establishing the appropriate control measures. Different analytical and diagnostic techniques are used to carry out the common monitoring of a farm. In general terms, serology allows us to detect and quantify antibodies against the Prrs virus. PCR is a molecular biology technique which allows us to amplify the viral genome and indirectly determine its presence and quantify it. Sequencing is a technique which aims to determine the order of nucleotides in a fragment of genetic material of Prrs virus. Serology gives us an idea of how the Prrs virus circulates on a farm. When interpreting the results, it is essential to bear in mind the serological technique and the commercial kit used, as the kinetics of detection can vary considerably. Currently, there is no serological technique that enables us to differentiate between vaccinated and infected animals. Bleeding animals at different ages, gilts, breeders, and growing pigs, will enable us to obtain the serum profile of the farm, which will provide information on its immunological and clinical status. Results obtained may be presented in 3 types of graph. Analysis of the results helps to determine the age of infection, the infection pressure of the herd, as well as the percentage of susceptible animals. This information is useful for determining the epidemiological status of the farm and applying the appropriate control measures. PCR enables us to detect the presence of virus in different types of sample through the amplification of its genetic material. In the breeding herd, it is commonly used to confirm the diagnosis of acute or subclinical Prrs outbreaks. In gilts, it is widely used to determine their status at the beginning and at the end of its acclimatization period. In growing pigs, it helps to determine its epidemiological status. These results will help us to apply the suitable control measures. Determining Prrs virus sequence is a useful tool for epidemiological studies. This technique enables us to create phylogenetic trees of field strains and to describe the degree of variation between strains. It can also provide information about the number of different strains present on one single farm. In case of Prrs outbreak, sequencing enables us to determine whether the strain causing the outbreak is the endemic one, homologous strain, or if it is the new one, heterologous strain, that has entered the farm. That's an important distinction, since corrective measures are different in each case. It should be taken into account that the degree of similarity of ORF5, or even the complete genomic sequence, is not a useful parameter for predicting the level of cross-protection of a given vaccine strain in front of a specific field strain. Results obtained from these analyses provide information on the epidemiological status of the farm. This will help us to choose the appropriate control measures and intervention strategies in each case. It is necessary to know the most appropriate biological samples to perform the analytical techniques and to know the type of information that can be obtained from each of these. Blood samples allow us to obtain results that are useful at individual level, for example, in process of gilt acclimatization. It is possible to obtain results for the whole population with sufficient number of samples of each animal group. Prrs virus has a tropism for a large number of different tissues such as lungs, fetus, tonsils, lymph nodes, umbilical cord, which are adequate for sampling. Some of these samples allow detection of the Prrs virus irrespective of the phases of viremia. Saliva is becoming widely used for performing both serology and PCR. Saliva sample is obtained through cotton rope, which is chewed by pigs. Sampling process is faster, cheaper, and non-invasive, reducing stress in the animals. Information obtained is for the whole population and enables better detection of a low prevalence of infection. Once monitoring has been performed, farms can be classified within different Prrs epidemiological status. This herd classification will help us to decide measures that have to be corrected and vaccine program strategy that have to be implemented. Evaluating the presence of virus circulation in some pigs' populations, in other words shedding status, and evaluating the serological and clinical status in breeders will allow us to classify the Prrs status of the herd. Herds with reproductive clinical and subclinical Prrs outbreak have virus circulation and high antibody level. Such herds belong to the Prrs positive unstable category. Those farms where the reproductive outbreak is being controlled but antibody level in sows are still high due to presence of virus circulating within the sow herd will be classified as PRRS-positive stable farms. Those farms where virus circulation has been eliminated from all sections of the farm will be classified as provisional negative stable in case they still have seropositive sows, or PRRS negative stable if no seropositive animals are present in the farm. In summary, monitoring will allow us to evaluate the epidemiological PRRS status of the farm. This herd classification, together with evaluation of biosecurity, will enable us to decide the best control measures to be implemented and vaccine strategies.